gotta figure out there's like a site license. If I were able to code things into a program and then tell you how to use them at home, so you can change parameters and watch what happens to graphs, is that something you appreciate doing? So last class or the test we changed beta. So I could code in an equation that looks like a hill equation, and you could just plug in numbers for beta and it spits out curves. Would you guys actually play with that? Like legitimately play with it?
Have you done on the mount?
What's that?
Have you done on the mount?
Yeah, I make it done on both. It's gonna take me the summer to put it together, but like, if you guys say no, we wouldn't do that, I just won't make it. I won't be offended. Would you guys actually use something like that?
Yeah, but we won't be in this class then.
Yeah, that's why I'm asking you. That's why.
Well, it depends, like, if you were gonna ask a question on a test, like, how is this, and this, and we had to be familiar with it, it'd be a really good tool, you know what I mean?
Okay. That's a good point. Would it help you answer, like, most of you guys did really well in that question, but would it help you guys answer questions like that?
Yeah.
Yeah? Okay, I can put something together like that. Okay. For this year, you're screwed. Alright. Let's talk about different levels that we can sort of look at biological processes in the cell. One way to look at transcription, so whether it's a transcriptional network or just the process of transcription itself, is using reporter genes. So I bolded this because this is one of the things that we're going to use, or we're going to examine in depth. So a reporter gene, what it's meant to do is report on activity of a gene at the transcription level. So we use a fluorescent protein, and the idea is that if you see fluorescence, it means that transcription of that gene is occurring. So we'll get into this a little bit more a little later on. There's different ways you can look at how much RNA is there, things like FISH, real-time PCR. Let's really not focus on RNA, because people kind of skip this step when they look at systems and synthetic biology. What they usually focus on are proteins, because it's the end outcome of everything. So you can look at how many proteins are in the cell by using fusion proteins, and whether you're using fusion proteins, you can use different instruments. For example, you can use a microplate reader. This measures bulk measurements of an entire population. You can use flow cytometry, which is where you can measure cells individually. Or you can use fluorescent microscopy, which you can either look at a population of cells or you can look at cells individually. So it's a kind of a combination between a plate reader and a flow cytometer. Then there's all these other methods that you guys covered in genetics, like Western blot, ELISA, mass spec. I will not ask you to reproduce this list. It's just to give you a broad overview of what's going on. And again, the ones in bold are the ones that we're going to focus on. When you pick a method to study your system, you have to ask yourself, what am I actually trying to study? Or what do I want to get out of this? So the first question is coverage. And what's meant by coverage is, are you studying one gene or one protein in specific, or do you want to look at the entire interactome or look at everything inside the cell? So this is the omics level. This is where you use genomics or transcriptomics, proteomics, things like, for those of you who did take genetics with me, did you guys do things like omics, like transcriptomics, RNA-seq, that kind of thing? All right, that's great. Okay, we'll leave it at that. But basically, omics means instead of studying one particular thing, 1, 2, 3, or 4, you're studying everything in the cell all at once. The next is resolution. Are you looking for a bulk measurement? Are you looking for the average of a population? Or do you want to go in and look at every single cell individually? Because both of these measurements give you completely different information. The next is a biggie. Invasiveness. Really, this asks the question, do you want to look at something that's alive or something that's dead? They both have pros and cons. Alive things keep doing a process. Dead things are fixed in time. Dead things are really easy to work with, but they give you less information than cells that are living. The next is accuracy, or what types of numbers you're going to get out of the cell. So are you looking for a yes or no answer, like yes, my gene is on, no, my gene is off? Or are you actually looking to go in and get a quantitative value out of this and say, look, In this particular scenario, this gene's on at 50%. In this particular scenario, it's on at 75%. The last one's also a biggie. It's a hot topic in the field right now, and that's timing. When do you measure your cells? Do you measure them continuously as they grow, or if you have to kill them, what time point do you take? Do you take 2 hours, 4 hours, 24 hours, 48 hours? What do you select in there to see what's going on with your dynamic? So the techniques that we're going to look at, they answer some of these questions. Some of them don't. Like we're not, because there's so many different degrees of freedom in here, we're not going to be able to cover everything. But we'll come back to this and sort of touch upon where each technique fits within these sort of methods. Yes?
Kind of unrelated, but you teach genomics.
No, I don't.
Oh.
I took it. I don't know what genomics is.
Oh, really? Okay.
I'll leave it at that.
So genomics is, there's different types of genomics. Genomics is basically bulk study of DNA. So you can study gene sequences, like A, T, G, and C, all the way up to how the genome is arranged inside the cell. What's the structural features of it? Because structural features kind of dictate whether transcription, translation are going to occur, that kind of thing. So you can measure it at lots of different levels. That's only DNA, because then there's RNA, there's proteins. Just, all right, big data, computers. I'm not going to ask any more questions.
I got an A, by the way.
All right. I don't know.
All up.
We don't need to discuss that. Okay. Let's first focus on DNA transcription. One of the things that's absolutely revolutionized the way that we study cells are fluorescent proteins. So if you think back to genetics, these are proteins where you shine a specific wavelength of light out into it, and it gives you a different wavelength of light back. And so that wavelength of light that it gives back will vary. So you can have blue cells, green cells, red cells, orange cells, purple cells, etc. Most of the time, reporter genes use fluorescent proteins. The reason is because you can physically look at the cell and ask the question, how much fluorescence do I have? So the question that you ask when you use reporter genes is, what's going on with my promoter? Is my promoter driving transcription, or is my promoter off? If my promoter is on, how much transcription is coming after it? So if you want to measure how much transcription is going on in the cell, one way you can do that is to make a fusion protein. And this is when you take your protein of interest, we'll call this POI. What you physically do is you physically attach some type of fluorescent protein, like a green fluorescent protein, to either the front or the back of this.
Wait, what's POI?
The protein of interest.
Oh.
So in sort of DNA world, this would look like this. Here's a promoter. Here would be, we'll just call this POI gene at this point, and you just clone in this green fluorescent protein at the back. This is a lot of fun and kind of cool to work with. There's bonuses to this. One of the major downsides of this is that you're taking two proteins and you're sticking them together. So there's a good chance that this green fluorescent protein is going to interfere with the properties of your protein of interest, and this might lead to a dead cell. At the same time, your protein of interest might interfere with your green fluorescent protein, which leads to a happy cell, but doesn't produce fluorescence. So instead, people said, look, this is an absolute mess. It's tricky. Usually if you have something, usually if you want to create a fusion protein, if you go to like a big tier 1 research facility, they'll take someone like Laura, and they'll say, Laura, you're going to do all her work. You are the fusion protein specialist. and that's all she does. You just bring her constructs, and that's what she makes. But nobody wants to do that job completely. Trust me. So instead, what people do is they construct a reporter gene. This is easier to do, less messy, takes 3 days. This could take months. Oh yeah, look at this wonderful image. So these are fluorescent proteins. Up at the top is the structure of green fluorescent protein. All of these things have this really cool barrel-shaped structure to them. So that's kind of the green crisscross on the outside. And you guys see that yellow part on the inside? That's the specific amino acid that's involved in giving off fluorescence. So people started with green fluorescent protein. They isolated this from jellyfish out on the west coast. And then they realized that green wasn't enough. So the original green fluorescent protein kind of lied somewhere here. And they said, look, why don't we just mutate green fluorescent protein to see what other colors we can get? And then, voila, we get everything from blue to plum. Really red.
Yeah.
Right? So we get everything from blue all the way to red. The nice thing is you can stick 2 or 3 of these into a cell and measure different parts of it. This is going to make it turn blue, this is going to make it turn green, etc., etc., etc. So then we've had, what I'll dub normal scientists go out and label the interior of living organisms like this nematode here called C. elegans. It's labeling certain proteins in it so you can tell where the proteins are. And then you have these wonky scientists that label monkeys with green fluorescence. These are like legitimate images. There's also fluorescent mice, there's fluorescent bunnies, there's a dog with a fluorescent nose. But most of the time we use these to measure cellular processes instead of making, weird looking animals.
You said it might interfere with the protein of interest. Would that like kill the animal or something?
If the protein of interest is essential, yes, then it would. To make the puppy with the glowing nose, what they did was they took a green fluorescent protein and targeted it to certain cells that show up predominantly in the dog's nose, and it would just cause its nose to glow green when it drank a certain type of water, and induce it.
That's awesome.
Pretty cool, right? But there's also this question as, why do we need puppies with green noses?
Was it just to like test the effects of the gene?
I really think that the way that publication went is, can we create a transgenic dog with fluorescence? As opposed to, can we actually look at something, scientifically rigorous? I think it was more of an engineering feat
Right,
than
like
anything.
to figure out if it works.
That's right.
Do one thing and then
Yeah.
does it not work.
So that type of research isn't done in the U.S. because the regulatory body, the NIH and the NSF, have said, look, let's not be really doing these giant transgenic animals. We really stop at mice. You can get fluorescently labeled mice that don't look fluorescents like this. You can get them. But as far as creating like bunnies and monkeys, zebrafish we work with, we've really kind of stopped with those organisms. We have fluorescently labeled human cells, but they're not like living. They're not like us. Okay, so we can use these fluorescent proteins to create reporter genes. All right, so here's the general idea behind constructing a reporter gene. Okay, what you do is you have a promoter. Let's call this promoter A. And your question is, I want to know what's happening with promoter A. When is promoter A activated? When is it not activated? And when it's activated, how much of it is there? Because it kind of gives you an idea, is if promoter A is regulating gene A, it gives you an idea of what's going on in gene A. For example, if promoter A is off, this is going to be off. If promoter A is on at 50%, you're going to have 50% output of gene A. The problem is that you don't really want to measure this directly. You want to use a surrogate. So what we do is we construct a reporter gene. So here, you take the exact same promoter. And instead of having gene A downstream, which you can't measure directly, it's not going to do fluorescence all by itself, you put a fluorescent protein downstream. So the idea is that when promoter A gets turned on, and it starts driving gene A, promoter A driving GFP will get turned on at the same time and the same amount, causing your cells to glow green. So, let's look at this. Does anybody have a favorite gene? Alright. Alright. Sonic the Hedgehog. The Sonic the Hedgehog is abbreviated SHH. And if you mess with this guy too much, especially in, well, you can't mess with it in humans. If you mess with it in mice, it starts to screw up how the mice develop digits. One of the genes does. So the problem is SHH is just a protein. It doesn't make any fluorescence at all. So in order to study this, you have to do a Western blot or real-time PCR, things that are a pain in the butt. So Sonic the Hedgehog is regulated by a promoter. Let's call that PSHH, promoter Sonic Hedgehog. So instead of trying to measure this directly, what you do is you take the promoter for Sonic Hedgehog and you put a green fluorescent protein in behind it.
And then the gene is after it.
Sorry?
Then the gene is after it.
Nope. So this would just be it. So it would just be a GFP. So realistically, depending upon the system that you're looking at, if it's in bacteria, this will be on a plasmid. If it's in yeast, it might be on an artificial chromosome. If it's in a human cell line, it'll be on a virus. So this will be independent of the gene.
So that gene won't be expressed anymore, just GFP will be?
Nope, so this is still inside the cell. But what you've done is you put in an engineered copy of GFP, added it. So
As
both,
well as that.
exactly.
Okay.
So this guy's still going to function as normal. You're still going to get digit information. But this is going to tell you when this thing is on. Okay, so let's say, for example, here comes the signal for Sonic Hedgehog. Was it like coins?
Yep.
We'll just call them stars. This guy here, what it wants to do is bind the transcription factor X for Sonic the Hedgehog, and it makes X star. Now the interesting thing about this transcription factor X, when it's bound to its signal, it doesn't care, and it can't tell the difference of what's downstream of the promoter. It doesn't know that that promoter has Sonic the Hedgehog behind it, and it doesn't know this promoter has GFP behind it. So, if you had two of these, one's going to go here, and one's going to go here. So the key thing is here, the transcription factor can't tell the difference between whether it's going to ultimately drive GFP or Sonic the Hedgehog. It's just looking for the promoter sequence. It doesn't care what's downstream of it. So what you end up getting from this, you get some Sonic Hedgehog protein, and from here, you get some green fluorescent protein.
Doesn't that have the signal, so half expression? Like, because you split the signal between two different modes.
Absolutely. So if signal and transcription factor are limiting, you will essentially have to signal between them. Most of the time, though, in these experiments, you put enough signal on so that it's not a limiting factor. So that all transcription factor is bound, and it shouldn't really be any different than
without the gene.
without the green fluorescence. But that is a limiting assumption.
But we can assume that there's enough sample.
That's right. Yeah.
And this happens at the same time that
Absolutely. So, if we think about this, what's nice about transcription, do you guys remember those silly ODEs I made you write out in the last test? All right. The nice thing about that is that in these two cases, okay, production is going to be the same thing because production is based upon RNA polymerase and transcription factors, right? So here, production is the same between both these guys here. The one thing that's a little different, and that really good papers will try to match up, but it's tough to do, is decay may be different. Right? So, the mRNA for GFP might be more stable than the mRNA for sonic hedgehog. Or, maybe the sonic hedgehog protein is more stable than green fluorescent protein. So when it comes to degradation, it's really tough to match these things up. But in terms of, I want to know if my promoter gets activated and it's turned on, this works out really well because production is the same in both cases. It's regulated by the promoter, it's regulated by the ribosomal binding sequence. Both of these things should be the same.
Don't you use the gene for the protein of interest as your like transcription factor, or the thing that makes the signal for the like GFP, your reporter gene?
So you can, but you have to assume that that particular protein of interest created the signal or would lock onto this. So this may not be a protein that either binds to DNA or it may not be a protein that has a downstream role that interacts with DNA. In that particular case, the setup wouldn't work because you need something to bind DNA to drive your reporter gene. In certain instances, if your protein of interest does create a signal, you can use a setup like this, but you're no longer measuring transcription. Instead, you're asking the question, how much protein is there? Because you move past transcription. Right? Because then what would happen is, this would make a protein, and then this protein would send some signal out, and then you'd have your GFP down here responding to that signal. So you're decoupling it further. There are reasons why you'd want to do this, absolutely. But for reporting on transcription, usually you keep the same promoter, try to make the ribosomal binding sequences the same, and you just sort of couple them together. Okay. So let me give you an example of this. Okay? So we've taken, some of you guys have taken these antibiotics before. Maybe not so much kanamycin, but derivatives of them. The way these antibiotics work is they interfere with the ribosome. Okay? So, if you interfere with the process of translation by interfering with the ribosome, eventually you kill bacteria, because the bacteria will want to go through cell division, won't be able to because they don't have any proteins anymore, and they'll end up dying. However, one of the things that the cells try to do to counterbalance the fact that translation is no longer working inside the cell is they think that there's something wrong with the ribosomes they've just made. So they sense this through negative feedback. Let's not worry too much about how that occurs. What they try to do is make fresh ribosomes that shouldn't have that problem. Okay? So, The antibiotics block up the existing ribosomes. The cell perceives this as being, I've just made faulty ribosomes, I'm not sure what's wrong, I'm going to try to remake some, and hopefully I get them right. So, at the bottom of this, when it tries to make more ribosomes, it increases the transcription rate of ribosomal protein, as well as ribosomal rRNA. It's incredibly challenging to measure ribosomes, because if you mutate or add a fusion protein just a little tiny bit, it kills the ribosomes in the cell, which immediately kills the cell. So instead, if you want to look at the transcription rate of ribosomes, you make a reporter gene. So instead of having Sonic Hedgehog, you might have a ribosomal protein, and this would be the promoter for it. So similarly, if you wanted to make a reporter gene, you put the same promoter here, but you keep GFP, or RFP, or CFP, or YFP, whatever fluorescent protein you have sitting around. And the idea is that if this increases in transcription, the cells will become more green. If it decreases, the cells will become less green. So here's an example of some specific data. This is time along the x-axis. This is green fluorescent protein. So it says normalized because what the authors have done is they've standardized it to the number of cells. So this is a per cell measure. They treated the cells here with kanamycin or chloramphenicol. And what they did was they took a ribosomal promoter and they put a green fluorescent protein downstream of that ribosomal promoter. And what they saw was that over time, the amount of green fluorescent proteins inside the cell increased. So what this means is that in response to either kanamycin or chloramphenicol, the amount of transcription from this promoter increased. You can measure that through green fluorescent protein. So this is sort of the types of data that you would see looking at a reporter gene. It's usually time, and then some measure of fluorescence along the y-axis. In this case, it's green fluorescent protein. Yes?
Did you say that the response to the antibiotics, the amount of transcription increased?
Yeah.
I thought you said before that it would stop ribosomes.
That's right.
So they're trying to make more.
That's right. Okay, so let's think about that, because I see what Austin's getting at. Let's draw this. Right? So what they're essentially doing is blocking this. And then what they try to do is say, okay, this is blocked up. Let's move back to this step here and go with this. So what the cells effectively do, because translation is blocked up, it's not like they can make more RNA polymerase. So what they'll do is they'll redirect RNA polymerase to promoters like PRNB. But whether they get a protein out of it or not, I don't know. We can't tell from this particular output because we're just measuring green fluorescent protein. The other thing that's a little kind of confounding in this is how can you get green fluorescent protein when translation's all messed up, right? So in this particular case, what the authors used was something called sub-inhibitory concentrations. So this is enough to slow down the cell growth. You're still messing with this, but it's not enough to kill the cell outright. So you're still getting the reaction without complete cell death. That's 20 micrograms per mil kanamycin, and that's 2 micrograms per mil. If you bump this up to 50 micrograms per mil kanamycin, this would be a black line because the cells would be absolutely dead. You bumped up chloramphenicol to like 5 again, flat line, nothing. Does that answer your question?
Yeah.
I still couldn't tell you what genome is. All right, let's look at something else here. So you can also make, actually before I move forward, are there any questions about the theory behind this guy here, reporter genes? Okay, so it's a non-invasive way to figure out the promoters on it. Okay, you can also use other types of proteins to report on activity. Usually people really like to use fluorescent proteins. In the past, however, people have used things like beta-galactosidase. You guys remember that blue-white screen with LAC-Z? Same thing here. So they would look at how much the cells turn blue as a measure of beta-galactosidase activity. People also like to use luciferase. This is a light-producing enzyme that was initially isolated from fireflies. The one downside of this thing is, so here is this chemical called D-luciferin, and what you have to do is you have to feed the system D-luciferin. So this is the substrate for this enzyme called luciferase. If you feed this substrate to animals or bacteria expressing luciferase, they give off light. The really nice thing about this is that a lot of the time the light is a lot more intense and a lot more bright than fluorescence is, so it's easier to measure it in things like mouse models. So these are mice that have different types of cancer, and what the authors from this did was they took the firefly luciferase, took the gene to encode that enzyme, and they put it downstream of a promoter that was responsible for either colon cancer, breast cancer, or skin cancer. And they asked the question, where do we see the most amount of light? Because that gives us an idea of how these different types of cancers metastasize inside the mouse. And so what they do is they kill the mouse, they put this mice in a box, and then they ask the question, where's the light coming from? So they also took out the liver, the lungs, the lymph nodes, everything else, but they could get an idea of overall, where's the most amount of light coming from.
And this is like a different thing than the fluorescent protein. It's like this to measure the same kind of thing.
Exactly, but it's a lot more sensitive. In a fluorescent protein, you have to shine in a very specific wavelength of light, and then you're looking for one wavelength of light coming out the other end. In luciferase, you could just make the mice drink some luciferin in their water. They don't notice the difference. Now instead of looking for one wavelength of light, you're looking for all wavelengths of light. So it's easier to pick it up inside the mouse. This is really cool, but the machine they use this in is damn expensive. It runs about $500,000. But you get a ton of information about this. Okay, so we're going to skip the RNA level. We're going to go right to protein. So we looked at how reporter genes, these report on transcriptional activity, if the promoter is on and off, but it really doesn't tell you anything about how much of this protein is there. It just tells you that the promoter's on. So one of the ways you can do this is a fusion protein. Again, we talked about this a little bit. It's when you have your protein of interest and you fuse your protein of interest to some type of reporter, like green fluorescent protein or red fluorescent protein. You can also use things like beta-galactosidase or luciferase. When you do something like this, people like to give this specific names. They'll call one the functional domain, the other one the reporter domain. The functional domain is your protein of interest as it functions inside the cell, hopefully doing its normal activity. This is the reporter domain because this is actually what's allowing you to see this, to report on the activity. So when you do this inside the cell, you would physically fuse these two together using DNA. So what you would effectively do is take out the stop codon from your protein of interest. The stop codon's gone. And instead of having another start codon, you just start right into the coding sequence of green fluorescent protein. So what ends up happening is the ribosome, once you've made your mRNA here, the ribosome hops on the front, and it translates this as one complete protein. The idea being that wherever your protein of interest goes, your green fluorescent protein is going to go as well. To give you an example, this is kind of like if you were, you guys ever see X-Files? You ever, not the new X-Files, but the old X-Files? Anyone? Anyone? One person? Did you see the one with the Carney? Who had like the evil twin that would pop out of the guy's stomach and kill people? That one's a good one. So the premise is there's this carnival man who's very fat. And he's not fat because he's obese, he just has another creature living inside him, and it would pop out and kill other people. So the analogy would be, this is the larger man, and this is kind of the freak that lives inside him. Now this doesn't kill anybody, and it doesn't leave, but the idea is wherever he went, no matter where he went in the US to run his carnival, this thing is just living inside and running around killing things. You can Google it. What's that?
Is that real?
No, of course not. It's the X-Files.
I missed the beginning. So I'm like, what are we talking about?
The X-Files? Come on. You guys ever watch this?
Oh my gosh.
Carnival episode. Here, let's see this guy. Yeah, see, there it is right here. This is the thing that jumps out of the guy's, they thought this guy here was the killer. He's eating like raw fish down by the stream. But it turns out it's not. It's really, I think it's this guy here. Yes, this guy here. He's got this thing living inside him, and this would kill him. No, it's like his twin. Oh yeah, he's all yeah, very much so. This is a giant fluke for those in parasitology that comes up through toilets and eats people. This is an unbelievable show that used to scare the crap out of me when I was a kid. All right.
Nightmares of the show.
All right, so this drags down wherever it wants. But this is the best measure of total protein that we have that's non-invasive. So we don't have to kill the cell to measure this because we can just say how green is the cell. The problem is, as I drew before, this can interfere with the protein's function, and the protein's function can interfere with whether it makes green fluorescence or not. So these are challenging to get to, but it's the best non-invasive way we have to measure protein, meaning that we don't have to kill the cell to do this.
So basically, after it makes the protein, it'll secrete GFP2, so you know that the protein's being made, basically.
Exactly, that's right. So that's exactly it. The other thing you do is like localization studies, where if this protein of interest goes to a particular organelle or a particular place inside the organism, that will show green. Okay, so just so you have this in your notes, again, the caveats of this is that the functional domain might interfere with the reporter domain, and the reporter domain might interfere with the functional domain. These are very challenging to create. So, we looked at sort of two different ways we can measure proteins using fluorescence, or we can look at promoters to look at transcription. The next question is, what exactly do you want to measure? It's okay to say I want to measure transcription or I want to measure how much protein is inside the cell, but what do you want to specifically measure? So what resolution do you want to measure this at? So one way you can do this is to take bulk measurements of the population. This is what's most often been done historically. Here what you do is you grow a population of cells, whether it's bacteria or eukaryotic cells, and you ask the question, how much total fluorescence is there in the population? With the assumption being, you're getting an average across the population. So a bulk measure is an average. It tells you nothing about the individual cells. It just tells you in general, is my promoter on or off and how much. One way to do this is through microplate readers. People like microplate readers because they can measure anywhere between 6 and 1,000 samples all simultaneously. You can also get dynamic measurements in these, which means over time. So you can say, I want to measure how much green fluorescence is in my cells as a population average every 20 minutes for 48 hours. And the microplate reader will do that for you. Microplate readers also read optical density. So how many of you guys have taken microbiology before? So when you look at one mechanism to measure how many cells are in solution is optical density at 600 nanometers. Yes? You guys remember something like that? So OD 600 gives you how many cells in the population. And then you can measure fluorescence. You can also measure luciferase. Some can measure beta-galactosidase. And some really fancy ones will take pictures of each and every individual well for you, so you can see what the population structure looks like. So here is a plate reader. This is my baby plate reader right there. A microplate are these microplates up here at the top. That top one there, that's a 96 well microplate, you can put 96 different samples in there. You put it into the microplate reader, and the microplate reader will keep track of how much fluorescence or light or optical density there is in each one of those wells. On the low end, you can get 6 well plates. A lot of people will do cell culture in these things, so if they're growing mammalian cells, they use 6 well plates. This is the type of measurement you get out of it. Again, here's time along the x-axis. Here's GFP, or this is CFP, so cyanofluorescent protein. This thing's been programmed to measure the amount of fluorescence in different wells on this every 10 minutes. So every 10 minutes, you get a data point. You get a reading of how much cyanofluorescent protein there is in your population. But again, you're just getting a population average. It tells you nothing about the individual cells that make up that population. All right. What I want to do is I want to stop at flow cytometry because this takes more than 4 minutes to explain. Are there any questions about what we've covered today? So the major thing really is fusion proteins versus reporters. And we looked at microplate readers. Yes?
If you only put like one thing before, like a set before, I can measure it.
Yeah, absolutely. You can tell exactly what you want to measure. Well, you wouldn't choose how much it behaves, because that's going to be programmed by the protein. You can choose when to measure exactly